Welcome to Adwoa Biotech, where we make biological sciences clear. Today, we're going to talk about an enzyme that lives a serious double life. I mean, on one hand, it's this total workhorse in microbiology labs, as well as a crucial tool for scientists. But on the other hand, it's also a life-saving medicine that's already working inside all of us. Are you ready for a game of Who Am I? I am a tiny molecule that acts as a bacterial test. Can you guess? Let me know in the comments. You want another clue? I'm used in experiments and involving RNA. Can you guess what I am now? Please let me know in the comments. If you need a final clue, I function as life-saving medicine. Can you guess now? Believe it or not, this wondrous molecule is a single remarkable enzyme. It's the common thread that weaves through all these biological activities. And we're going to follow that thread to some pretty incredible places. Our whole story kicks off in a place you might not expect, a microbiology lab. Looking at a really simple but super clever color range test. Okay, so check this out. To see this enzyme do its thing, scientists have this really clever test. They grow bacteria on a special petri dish that's got DNA mixed with a green dye. Now, if you grow a pretty harmless bacterium on it, like Staphylococcus epidermis, not much happens. The dish just stays green. But if you put its more dangerous cousin, Staphylococcus aureus, on there, you get this clear halo. This little see-through ring around it. And that halo, that's your first big clue. It tells us that this bacteria is pumping out an enzyme that's literally shredding the DNA in the dish. This is called the deoxyribonuclease test, or just DNase test for short. The enzyme doing all the work is DNase, and it basically acts like a pair of molecular scissors. Its whole job is to hydrolyze DNA, which is really just a fancy way of saying it uses water to break it down. When DNA starts chopping up those DNA strands, the green dye has nothing to hold on to, so it just floats away and the color vanishes. It's a really simple, elegant way to see if a microbe has this special power. But trust me, its job as a bacterial ID is just the beginning. So scientists saw this DNA shredding power and had a light bulb moment. They realized this could be incredibly useful. A really useful tool. That simple bacterial test was about to unlock one of the most essential tools in all of genetic research. You see, for scientists studying RNA, that's the molecule that carries the instructions from our DNA. Remember, the exon gets copied and it leaves the nucleus, comes out, the messenger RNA. Yes, so RNA. So getting a pure sample is absolutely everything. Like this quote says, even a tiny bit of DNA mixed in can completely ruin an RNA-focused experiment. It can make the results confusing or even just plain wrong. It's kind of like trying to identify who is whispering when there are two people in the room, but they have their back to you. So how do you get rid of the whisperer you're not interested in this case DNA, when you're wanting to focus on RNA? Well, scientists had a brilliant idea. What if they could borrow the exact enzyme from the bacterial test? They needed a tool that could surgically remove the DNA without touching the RNA. They needed those molecular scissors and DNase. It was the perfect tool for the job. And the process itself is beautiful in its simplicity. You start with your purified RNA sample, which may be mixed with DNA. It's hard to avoid DNA when you're extracting RNA from the cell. Then you just add the DNase enzyme, our molecular scissors. The DNase goes to work specifically targeting and chopping up all the DNA, but leaving the RNA totally alone. And what you're left with is a pure RNA sample ready to go for super sensitive experiments like reverse transcription, quantitative PCR, which is used to measure how active our genes are. It's just an elegant solution to a really big headache in the lab. But this is where the story gets really good. This incredible tool wasn't something that was invented in a lab. Nope, it was discovered. We're about to look at its original and most important job. So, you have to ask, right? Where did this perfect biological tool come from? If bacteria can make it and scientists are borrowing it for experiment, where did nature come up with it in the first place? The answer is kind of mind-blowing. It's not just in bacteria. The service rendered by DNase is a fundamental part of our own biology. It is literally flowing through your body as we speak. Inside our bodies, DNase is the ultimate housekeeper. It helps to digest the DNA in the food. You eat food, the proteins, the carbohydrates. There's DNA, nucleic acids, and DNase does the job of digesting it. But its most critical role is cleaning up after our own cells. Every single day, billions of our cells die off as part of a totally normal process. DNA is the cleanup crew that swoops in, chops up all that leftover DNA, and prevents it from piling up. This constant cleaning is so important because it stops our own immune system from getting confused and attacking our own DNA as if it were a virus. And this housekeeper is everywhere. Our pancreas pumps it into our digestive system. It's in our saliva, it's working in our kidneys, our intestines. This isn't some rare specialized enzyme. It's a basic part of our body's operating system, just quietly working 24/7 to keep things running smoothly. So we've got this amazing essential system for cleaning up DNA. But what happens when the housekeeper doesn't show up for work? It's a really important question. If cleaning up all this old DNA is so vital, you can bet that the consequences of failure are pretty serious. And you would be right. When that housekeeping system breaks down, bits and pieces of our own DNA just get left floating around. Now, our immune system is trained to attack foreign DNA like from a virus. So when it sees this leftover debris, it gets confused and sounds the alarm, launching an attack against what it thinks is an invader. This friendly fire on our own body, that's the very definition of autoimmune disease. And this isn't just some theory. There is a direct proven link here. Scientists have found that people with certain autoimmune diseases like lupus very often have faulty or just not enough of these DNase enzymes. Their internal housekeeper just isn't doing its job right. And that leads to buildup of DNA junk, which can trigger that whole devastating immune reaction. This brings us to the final and honestly the most incredible part of this story. We've seen DNA as a bacterial marker, a lab tool, and a crucial part of our biology. Now let's see it as medicine. And this is where all those different threads we've been following finally come together. The knowledge we got from a simple petri dish, combined with that we learned about genetics and our own immune system, paved the way for a truly revolutionary medical treatment. And you can see this breakthrough perfectly in the treatment for cystic fibrosis. So with cystic fibrosis, or CF for short, the lungs get clogged with this incredibly thick, sticky mucus. And it turns out a huge reason for it being so thick is because it's jam-packed with DNA from dead immune cells. A massive failure of that internal cleanup crew we were just talking about. So the solution, it was brilliant. If the body's own DNase can't keep up, why not just give it some help? That's where Darn Alpha comes in. It's a pure lab-made version of DNase that patients can actually inhale. It goes right into the lungs, acts like a supercharged housekeeper, and just starts chopping up all that tangled DNA. The mucus thins out, and suddenly it's so much easier for patients to breathe. Just amazing. This story of DNase is just the perfect example of how understanding one fundamental biological process can lead to these powerful new therapies. It started with a little halo around some bacteria on a dish and ended with a life-changing treatment.
Be strong.